It's a beautiful day in the neighborhood, is it not? Welcome, everybody. So, I should have prepared better. Gotta get some tabs open here. Tomorrow, as you can see in the thumbnail, I am debating Sal Cordova. I don't know if any of you have seen his debates before, but yeah, there we are. Evolution versus intelligent design. I've only ever watched one of his debates, and it was he debated Grayson and somebody else alongside this chick named Taylor about I'm pretty sure that was Origin of Life. So if I just type him in, how many times does he come up? Creation versus evolution. Oh man, he's been on here several times. Where's the... No, that's not the one I've seen. Where the heck is the... Man. Oh, geez. He went up against Aaron Ra. Oh, yeah, this is the one, right? Yeah, this is the one right here. The thing about Sal, it's always funny in a lot of the videos I've seen, his face is always like so close to the camera that it just... It's, you just can't see hardly anything. So I'd say biogenesis, but apparently his thing is evolution. So we're gonna do some opposition research today. Creation. What is this? What creation evolution headlines? Sal Cordova has appeared on national TV, radio shows, newspapers, books, and magazines for his work in promoting intelligent design. Ooh, which isn't a thing, and creation science. He's a former scientist and engineer in the aerospace and defense industry. Absolutely zero relevance toward evolution, and presently serves as professor and researcher in the area of Christian apologetics at a small Bible college, although we won't ever know which one that is, apparently. Through the generous support of a private foundation, he also manages a small privately held investment fund, whatever. He has degrees. This is really funny. On April 28th, 2005, that's my mom's birthday. That's kind of funny. He became one of the few creationists to ever appear on the cover story of the prestigious scientific journal Nature. So I looked that up, and it's, I mean, yeah, I wouldn't exactly call it a hit piece, but it's kind of a hit piece. So this centers on the whole intelligent design controversy in the what was that? It was Kitzmiller versus Dover Area School Board. So that comes from this. That was the what do you want to call it? The what the hell do you call this? The trigger warning that they were forced to read to all the students before they started teaching evolution. There's a really good Nova documentary about this. I think it's called Intelligent Design on Trial or something. Yeah. So I'm pretty sure on the PBS website, I think you can watch the whole thing for free. Yeah. Well, I have before. I know that I have. I remember doing it, but it was a really long time ago. Watch that documentary if you haven't. It's pretty funny. But yeah, I mean, the whole article is titled "Dealing with Design," or in other words, putting up with it. The idea of intelligent design is being promoted in schools rather than ignoring it. Science need to understand its appeal to help students recognize the alternatives. Make this a little bigger so I can read it. Has recently become popular amongst Christian fundamentalists to research as it sounds like politics rather than science and like somebody else's problem. Mixing as it does the supernatural with scientific doctrine, the concept is a throwback to the days when natural philosophers pursued pseudoscientific disciplines such as alchemy. Professors rarely address the conflicts between faith and science in lectures, and students are drawn into ID as a way to reconcile their beliefs. In doing so, they're helping it gain small but firm foothold. This is bad news for researchers. Yeah, I mean, this is not a flowery review. It's which isn't a thing, proceeds not to prove it. Well, it was proven in court that it's not a thing. But we'll talk. Wow, your mom's young. No, she wasn't born in 2005. She was born on April 28th. That's what I meant. So what can scientists do? The concepts advocates frequently approach researchers with debates, so on and so forth. Yeah, what is their advice? But attacking or dismissing intelligent design will likely aggravate the rift. Scientists would do better to offer some constructive thoughts of their own. For religious scientists, this may involve taking the time to talk to students about how they personally reconcile their beliefs and their research. Even if they manage to sway just a few students, researchers in the United States can have a disproportionate effect on the national debate over science in the classroom. Yeah, so I mean, it really doesn't say too much, I guess. And then, oh shit, it went away. So I don't know exactly what we're going to take a look at, but we're just going to search up Sal Cordova and see what kinds of things we have on him. Sal is slimy and aggressively obtuse, yet seemingly polite little turd all at the same time. Yeah. Really need you and Professor Dave to hop in a live stream and debate Flurf's young Earth creationists. That would be kind of funny to do. I feel like maybe we would make a good team. He's a lot more aggressive than I am. Oh God, Sal is horrible. Yeah. Oh, before we do this, Kitty Cat, you're gonna have to get up for a second. So I changed my picture behind me. I don't know how many of you know this, but my dad started painting years and years and years ago. So I'm gonna try on average about once a month to change the picture in the back. And I never really showcased the last picture. The first time I put one up there, I had like a picture of it for everybody to look at. But this is the one that's been up there. I think you guys can see here. I'll tilt it so the ring light. This is the one that had been up there for a little over a month. Some pronghorn antelope. So that's what my dad painted back in. I don't see the date on this one. Is it on the bottom? I don't know. The date's on it somewhere. Who knows? But for now, it's you can't really see it very clearly. But this is one of my favorite paintings. It's just really cool. So good job, Dad. Happy trees. My dad doesn't really paint happy trees, but I guess we can call them whatever we want. So I only just for 30 seconds took a look at this before the stream started. So, but he actually has these hour-and-a-half long conversations, not even debates, with I get information from these two all the time. Creation Myths is a small channel, but he has really good stuff. Protein Orchards and Universal Common Descent.
Hello, everybody, and welcome back to Creation Myths. We are here tonight for what I think will be a really fun and, if history is any indicator, probably just like a really nerdy show. I'm really excited. I've got Sal Cordova here with me, and we are going to talk about protein orchards, just to say the separate ancestry of different protein families and the implications of that for evolutionary theory.
I mean, if they're going to talk about shit that I don't understand because I don't have an advanced degree in this, then this won't actually be all that helpful. But I do worry that Sal will have something really bizarre to bring up. I'm like, I've never heard of that before, but whatever.
Before we get going, let me just take a second here and tell you that Sal is an old hand in the evolution, creationism, ID stuff. He's been doing this for...
Oh, by the way, Dr. Dan, when there was a poll of graduate creationist science students, biology students. They said, who would they rather have, Dr. Dan or Kent Hovind? It was Dr. Dan unanimously. And here is Dr. Dan correcting me on something with Jeff Tompkins. Oh, you wanted to say
I just want to say, I should hope so.
Dude recently came on my channel like 2 weeks ago. He said, Sal, you're so much better than phylogeny of cars. And we'll talk about that because that relates to protein orchards. I adjusted this talk today just because of that comment. I said, Dr. Dan's right. So I came up with it. And we'll talk about Owen's homology. In our debate 4 years ago, this graph was prominent. I said, Dr. Dan, I'm going to have to work on that. And so, and there were it led to genetic entropy 2.0. And
geez. Genetic entropy is so, I wrote about it extensively in my book, so I'll have to reread my book to have all my talking points and data points in order.
Dr. Dan had confronted both Michael Behe and Otangelo Grasso, and I said, I think Dr. Dan's right. Irreducible complexity needs more qualification to be effective. So, by the way, Erica is also one of my favorite evolutionists. We may or may not have, if we have time, I actually presented on her channel. It wasn't a debate. It was just a friendly discussion about molecular phylogenetics, and that may have some relevance to what we talk about today. You can take that down. So I just wanted to say I'm very appreciative. This has been I think
way back in like the Panda's Thumb and Uncommon Descent blog wars days. So we go way back. So Sal, the topic tonight is this idea of protein orchards, and I've been wanting to talk to you about this concept because if I recall, you gave a talk at a recent creation conference on this topic, and basically you're the guy for this argument. Like if you want to talk about irreducible complexity, you talk to Michael Behe, right? If you want to talk protein orchards, you're the guy making this argument. So I'm really happy to have you on to be able to kind of work through this. And I will say this, you've been making your rounds talking about this. So Real Science Radio has you've been doing some interviews with them. I've watched the recent ones. The most recent one that they posted was on this exact topic. So I want to hear you make this case. Take 5 minutes, 10 minutes, make this case about protein orchards and evolution, and then we'll talk about it. Everybody,
I'm thinking that might
great.
be what
I
this
actually
is
think
about.
We might get
Sydney.
some agreement. And Dr. Dan, I made a presentation at Liberty University Creation Research Society, and I actually featured a video of Dr. Dan. I spoke of you, reverently, just so you know. I usually don't speak reverently of most evolutionary biologists, but I do of you. Just
get so
Joe
close to the
Felsenstein.
camera.
So if we could bring the slides up again, this relates to peer. How some of this developed was I was working on various publications. I recently, and Dr. Dan gave me permission, just a little bit of self-promotion. I published a peer-reviewed research article as a co-author in Oxford University Press, and some of the stuff that is in that article was kind of in the area of research, structural bioinformatics. It kind of motivated what I'm, my thesis tonight. One of the co-authors is Joe DeWeese. He's published in the journal Nature. He and I also published in the Creation Research Quarterly. We published like in FASEB. And then also some of what we do tonight will actually relate to a Springer Nature reference chapter on fitness maximization. This is probably where Dr. Dan and I will disagree, but let's go to where we might agree, just to clarify some things of what I'm saying and what I'm not saying, because it's been mischaracterized on the internet and it's hard to get people to untangle things. And I understand the reasons why. I just want to point out there are people in the ID community that are ambivalent or they believe in common descent. Michael Behe said he accepts common descent. Richard Sternberg, he's still in evolutionary biology. He probably accepts common descent. Michael Denton. So these are names you might be familiar with. I'm actually, for the sake of this talk, going to say, let's assume common descent, organismal common descent. But that's different than the descent of all proteins. Now, this was just from a debate. This is really funny. Aaron Ra wrote this to me, and he consulted you. And I said, look, this is something I think we're actually all going to agree on, that proteins/genes do not have a common ancestor. So again, I want to say this is not a criticism of universal.
I find that to be a little bit odd. I'm not 100% sure what they mean by that, but okay.
Universal common ancestry. Absolutely not. Now, people know I'm a young earth creationist, but I think this is the wrong argument to use for young earth creationism. And I actually think some of the evidential case for young earth creationism isn't where it needs to be. And so I just want to be on record. That's why you don't hear me talk about it that much. I think Dan's side of the argument, I would have a hard time defeating it. So I'm just going to assume universal common ancestry. Also, I've been accused of saying that's what evolutionists believe, that all proteins descended from a common ancestor. I'm like, no, I never said that of anyone currently. So I'm not accusing anyone of saying that. but it's kind of aggravating that I get mischaracterized. I'm trying to just set the record straight. There are not many Google hits or evolutionary papers that I find that talk about the protein orchard. So evolutionary biology is more than the assumption of common descent and inferring phylogenies. Here's some example titles where you could actually explore the questions that are active. So I'm not criticizing common descent, and I'm not criticizing phylogenetic methods as being valid or invalid. I'm talking about the mechanisms of evolving proteins. And these are some papers that Dr. Dan may be familiar with. There is the word homology, and currently homology, and Dr. Dan probably understands this better than I do, it's very tied to common descent. That wasn't the original.
Okay, we got to get to something that's a little more interesting. But I actually forgot to mention this. This is the Atheist Experiences. It's like a sister channel of theirs, Truth Wanted. So Kelly Laughlin and I did this like a month ago, and Kelly was also the co-host for that. So catch me on there tonight, or don't, whatever.
In a model where you have a organismal common ancestor, but then independent protein families. So we're in total agreement there. So maybe we could quit because we're actually in agreement.
Good. Quit while you're ahead. Well, I think it's the next step is where I'm going to disagree. So, okay, if you want, I can pull the slides back up, and then I think it's the next bit. That's where I'm going.
My thesis then.
So here you go. Keep going.
All right. So I say that the creationists are very quick to invoke supernatural miracles or metaphysical miracles. I'm just going to say, okay, let's set aside these philosophical things. We could at least say in principle, from a statistical standpoint, what events we would consider like very, I wouldn't call it a miracle in the medical physical sense necessarily. I'll just say astronomically far from normal expectation. So if you have 500 coin, if you have a flip a coin that's fair and it's heads 500 times in a row, that would be astronomically far from normal expectation. Whether it's a miracle of God or not, that's different than the actual statistics of it. Separate question. This relates. We are dealing with the question of evolutionary fitness, and really in the last part, because most of this was about Fisher's fundamental theorem of natural selection. But at the very last part, we are questioning fitness, and that relates to evolvability, how well, effectively, Darwinian so-called natural selection can evolve something.
Yeah, I mean, he's not saying anything that is beyond comprehension so far. He just hasn't said anything yet.
And it was after we published that I started to find all these other papers like selection-driven gene loss in bacteria, mutated genomes decay despite sustained fitness gains. I was seeing a decoupling between, I would expect that natural selection should be building genes. And I mean, that was the way I originally understood the theory. But as I studied population genetics, I said we ought to see a lot of loss. And I began to see experiments to that effect. You've done a video about me redefining fitness. And I just wanted to say, I'm not the only one that has. It's not just the creationists that have made the objections. I probably went farther than the evolutionary biologist. Lewontin's very respected. This is a great article, The Confusions of Fitness. I go farther than he does, though. And Andreas Wagner, also a very respected evolutionary biologist, he said, fitness is difficult to define properly. Taken together, these difficulties mean an enunciable measurement of an organism's fitness. Organism's fitness does not in practice exist. And I'll go into that a little bit more.
Can I just say on that, like, I disagree. And like,
okay.
Yeah, what I basically say is fitness is a function of the rate of reproduction. If it's steady, then you have good fitness. If it's increasing, you have even better fitness. If it's decreasing, then you have poor fitness. So it can literally just, like, if an organism doesn't fit in its environment, then it's not successful. And you measure that by the rate of reproduction.
Evolutionary biologists like disagree with each other. I disagree. I think it's pretty easy to measure. I mean, it's not necessarily easy in a technical sense to measure fitness.
Wrong, Sydney. There's a final possibility. The coin is flipped and it spontaneously cleaves right down the lengthwise and then lands both heads and tails.
But like, in principle, it's not a hard thing to do. Fitness is one individual's genetic contribution to the next generation relative to the other individuals in that population. That's your fitness. And
okay. So Dan's measuring fitness at an individual level and saying, the alpha wolf has greater fitness than the other members of the pack by virtue of how much genetic material it passes on. Interesting.
And like, you can measure this a bunch of different ways depending on the system. Like for me, looking at viruses, like all my grad school work was experimental evolution of bacteriophages, viruses that infect bacteria. And when you're dealing with viruses in a petri dish, you measure doubling time. So I can look at different populations and measure doubling time, and I could actually compete them against each other and see which ones would win. And like, it's really easy to measure fitness in that context. You figure out how to do it. You do the you do the math, and like you can do it. And depending on the organisms you're looking at, measuring one subgroup's or one individual's relative genetic contribution to the future compared to other individuals or other subgroups can be a hard thing to do. Like just in a technical sense, that's a hard thing to do in many cases. But like in principle, that's what fitness is. And I'm sure there are obviously biologists that disagree with that definition of fitness. But like you take what as defined by Darwin and you update that definition for modern molecular genetics. That's what fitness is. It's you're using looking at allele frequencies and such, and what's your contribution relative to everybody else, right? That's what that is.
So thank you for pointing that out. And I actually, in this is a paper from a smallish, ID-friendly journal. You could see that I actually go through kind of like what Dr. Dan was describing, but also kind of the formalisms of how it's traditionally done. Now, I just wanted to point out something. Andreas Wagner, again, in his book Robustness, he said, however, fitness is hard to define rigorously and even more difficult to measure. An examination of fitness and its robustness alone would not yield much insight into the opening questions. Instead, it is necessary to analyze on all levels of organization the systems that constitute an organism and that sustain its life. I define such systems loosely as assemblies of parts that carry out well-defined biological functions. And this is very similar to Michael Behe, a single system composed of several well-matched interacting parts that contribute to the basic functioning of the system. And I'm gonna show why I think that the standard, let's call it the standard definition of evolutionary fitness, does not actually solve the problem that Darwin is trying to solve, which is the emergence of organs of extreme perfection and complication, Origin of Species, chapter 6. So again, we have, like you said, the doubling time. That's an exponential function, and you can relate that to absolute fitness. You can scale it. This is in my Blythe paper. It's an educational paper. It wasn't meant to be a research article. The problem that Lewontin articulated in the Santa Fe Winter Bulletin, and one evolutionary biologist said when he read that, he rejected Darwinian evolution from that point on. And I'm just extrapolating a little bit because Lewontin had this density-dependent fitness. He said, well, then what is fitness? Because if the environment changes, actually, Dr. Dan, just really.
Okay. I don't have the patience for whatever that was. The interviews, I might watch those on my own when I can't sleep at night or something. But let's see. What do we got? Evolutionary fitness pro, because that's a very, very specific topic there. Evolution on trial. I don't know who those people are. The Big Bang on trial. Oh, geez. This will probably be a shit show, but we're going to check out that. Why would you debate Pascal's wager? That's such a weak proposition. Atheist Junior really knows his shit, so we'll check this out.
The appellations, by the way, there are.
Who the hell is this Cindy chick? Cindy Lou who? Well, let's listen to Sal's opening statement to get an idea for maybe what direction he'll go in.
But not between different genera, even in the same.
So on the creationist side, they believe that not elsewhere.
He could only means a thing to not remote influence.
Geoset forms off the in order
about
to
whatever
design by
membrane bound, ordinary involved in double-stranded eukaryotes.
Viruses can be God rather than assuming common descent with no God. Take it away, Sal.
Hi, kitty cat.
And thank you, Cindy.
You have to wave.
Some of the problems that I will point out, we can discuss it in the free discussion, are problems with the transition from prokaryote to eukaryote. I deal with diagrams like
well, that's easy.
This in the origin of eukaryotic chromatin. And this is just all the machinery involved in double-stranded break repair for eukaryotic chromatin. The number of simultaneous events and things that have to be there for this to work just boggles the mind. If not in the theological sense, it would be a miracle in the statistical sense. Gradual pathways do not at all seem reasonable, and that is subject somewhat to experiment. There's also the problem of the membrane-bound nucleus. of the eukaryote, this would involve just so many things that it just boggles the mind in terms of the complexity. There would have to be the membrane-bound organelle plus all the ports, like the nuclear pore complex, the import-export machinery involved, the gatekeepers, and then things called nuclear localization signals. I have diagrams on that, and also translocation processes. I've had the privilege to work with Dr. John Sanford. He is a Cornell research professor, retired. His invention, the gene gun, has fed starving billions. He represents the unsung creationists that you don't hear of. He is published in all sorts of venues, including the prestigious scientific journal Nature. He published in the Journal of Mathematical Biology. I've had the privilege of being his colleague and worked for him. This theorem that they published set records in the Journal of Mathematical Biology. I have then worked with them on the population genetics of this. Natural selection does not work according to what you think it does in the textbooks. The elite know that it's kind of a failing paradigm, and we can cover that. He was invited to the National Institutes of Health to talk about this problem. We are decaying. And there are all sorts of other things. There, as far as mechanism, we do have on the table by various physicists that God exists as a possibility. And I would just say that...
what does this have to do with evolution?
Argument by contradiction is not an argument from ignorance. How improbable does something have to be? How complex before we begin to entertain the possibility that it was a divine miracle? We can scientifically argue from probability. That's well within the bounds of science. If we're willing to be speculative, we can invite this conception of God just straight from quantum mechanics without appeal to any sort of theology. This leads to testable predictions about the human genome decaying, the destiny of the biosphere, the destiny of civilization. So I look forward to the free discussion, and I want to thank everyone for joining.
That was his entire opening statement? 24:46 to 28. So it was 3 minutes long.
Yes, tonight.
Too rowdy. Sorry, go
Sorry.
ahead.
Handshakes across.
So I thought, basically to predict that these. So with that, then.
Yes, and I would just say this: in geocentrism, you had testable predictions that were fulfilled. It was the anomalies that broke it. So in geocentrism, you could predict eclipses and all sorts of other things.
So would that then suggest that? Sorry, would that then suggest that your hypothesis that these model organisms are well suited for discovery of like health issues in humans? Would there be anomalies there that would disprove that hypothesis? How would you go about disproving that hypothesis?
Oh yeah. Okay. So there is a peer-reviewed publication that I've worked on, and it's actually been pioneered by a lot of other people on this topic. Where we so first of all, since I knew you're a neuroscientist, I put the squid up there for you. I knew
appreciate that.
Significant.
Yes, they're
those up there. Neuroscience was pioneered by the giant squid axon. The squid is not very, as far as I know, very near us, but it's beautifully optimized in some sense to enable it to probe its nervous system, because we could insert little probes in it because it's just so big relative to other creatures.
I would say that it's because they don't have myelin. That's why their axons are so big. So they never evolved myelin. That's something that's specific to vertebrates. And so they don't have any myelin sheathing. And that allowed for those axons to be very large, which makes it easy to record. And that's why early studies to identify how action potentials
work. Are you sure that's what we're going to see?
A lot of that work was done in the giant squid, like you pointed out.
I started working on, I'll just show you guys. Sal's not going to see this. You know what? Maybe he will see it. So, I mean, I don't think so. But I only prepared 5 questions because I emailed James and I said, well, can we have cross-examination for the debate structure? And he said, I'm willing to do 10 minutes each side. I was like, 10 minutes? Which I didn't have to prepare much for that, I guess. But I only came up with 6 questions. But yeah, my final question is, can you give me any predictions that intelligent design makes? And he won't. So, I mean, we'll deal with that in real time. I think it'll be pretty funny, but yeah.
Okay, if I could share my screen, maybe I could more directly answer your question, Doctor. Again, we have similarity. I'm not denying that of the human and zebrafish. That's the collagen. And we can say it's 75% similar depending on your scoring technique. So, and then you can build these nested hierarchy diagrams, these phylogenetic. It looks like compelling evidence for evolution. But then I show that there is the problem that the proteins don't have really good, there's not a common ancestor like here. So on the left is collagen, on the right is the zinc finger 136. And like on the left, the glycines are highlighted. On the right, the cysteines and histidines are highlighted. And you can see it's a very distinct architecture. So there's no common ancestry in the proteins. And I think that's problematic, and it's also a probabilistic thing. But to the point of biology being optimized, one project I'm working on is if you take the sequences like say zebrafish, humans, and you take the homologs across a large spectrum of species, you put it through an algorithm that looks kind of like this here. You can look it up on Wikipedia. It's the direct coupling analysis. It can then give you the protein fold, the three-dimensional. It's just like voodoo. And to me, I mean, just it's worth exploring. But the testable hypothesis is if we start to remove creatures from the set of homologs, that the protein fold prediction will fail. That would indicate to me, as I was saying, the hypothesis we put forward, this is biology is optimized for scientific discovery, that both morphologically and then also at the molecular level, it's optimized. But you really see it with this. And if one is a young Earth creationist, you'd have to say, okay, why do you have all the similarity there? What's the pattern? Because it does look evolved, and we've acknowledged that here. But if it's young, that would be kind of the ugly fact that destroys evolution, that it's not common descent, it's common design. I don't know if I answered your question.
Yeah, I mean, I'm actually struck by a story that I know quite well, which is the discovery of the Parkin gene. I don't know if you're familiar with this, but it's called the Parkin gene because this gene, the first thing that we learned about it is that in humans that have a specific mutation in this gene, they have early onset Parkinson's disease. So we identified this gene. We said, oh, well, this must be the gene for Parkinson's disease because we had no idea what it was involved in. So eventually, though, what we did is we went to the mouse model, which is what we usually do. And so for those who don't know, Parkinson's disease is a specific disease where you have a loss of dopaminergic neurons in the substantia nigra pars compacta. And
duh, everybody knows that.
That loss of dopaminergic neurons means you have insufficient drive to allow for planned motor behavior. And so then you can no longer, you're frozen and stiff. So why not do this in mice, right? Because we can't do it in humans, like you said. It's just unethical. So we're going to do this in mice. We're going to knock out the Parkin gene in mice. And what we find is that there's no Parkin phenotype whatsoever. Now, they do have some effects, but it's mostly has to do with like weight gain. So there's some changes in how they deal with high-fat diet and also a couple of effects on the heart. But they show absolutely no effects on Parkinson's disease. And it turns out that the Parkin gene is involved in a process of what we call mitophagy. So that's how we get rid of damaged mitochondria. Now, it does it both in humans and in mice, but the ultimate effect of knocking out this entire gene in mice leads to basically no phenotype, no Parkinson's disease. And there were millions and millions of dollars wasted on grants to identify this. Of course, they discovered interesting things about how Parkin works, but it completely violated the hypothesis that, wow, we're going to have now a way of looking at Parkinson's disease in mice by knocking out this gene. And it just didn't work at all. And there's lots of examples like that.
I don't know what he's talking about, but do you guys remember like a couple of years ago, I think it was actually Alzheimer's research. They kind of, I don't know if they proved it, but it seemed like they proved that the whole thing about, what is it? Not the plaques, but something about Alzheimer's. All the research that's been done over the last 20 years was based on a paper, and they looked at the paper and like, this person faked all the data. And so there's this chance that we've spent like $1 billion on Alzheimer's research, and it was like literally all for nothing.
Mice are great for lots of things, but oftentimes we find that there are important subtle differences that just completely illustrate the fact that we have to take into account that they are not tiny little humans that were perfectly designed for us to allow us to discover interesting things about the way humans work. And I mean, it just kind of blows that whole hypothesis.
Okay, I just want to listen to what Sal has to say.
As much medical technology, if we didn't have model organisms, I'm not saying that they're perfect, but they certainly have immensely helped. I can't imagine us doing medical technology and advancing our understanding of human without model organisms. That's why we have such a suite of them. And the alternative is then to be dissecting each other. So I'm not saying that they're perfect models, and that's why we call them model organisms. But that being said, in terms of pure science, just as a scientific question, and that's why we try to subdivide this, where we assume common descent. It's like, okay, we can at least argue complexity. But then, we would have, I'm just conceding to you that with an old Earth, and you look at the pattern of biology, it's hard to deny common descent. So I think we're on agreement there. And I've tried to tell creationists, don't argue that way, because if you're going to argue old Earth, and you try to argue against similarity, you're going to lose. I do gene browsing a lot. I could see genes that are 100% identical between us and chimps. I also see the patterns of pseudogenes, and I've dealt with allele elements and ERVs. So, guys, just creationists, if you're listening, don't go there. But if the Earth is young, and I have gone from being a theistic evolutionist and an old Earth creationist to a young Earth creationist, some of the stuff in physics has started to make me think, and enough that it put enough doubt that I said, just given kind of my own,
what?
Because I'm a Christian and I have. I read the Bible. It's like, okay, it seems enough of it aligns with the way that I see the scientific evidence. Just as a matter of faith, I said, I'll accept it by faith. I'm not going to pretend I have the science. But there have been certain things with radiometric dating that have only emerged like in the last year with heavy electron quasi-particles. There may be a pathway to changing nuclear structure, and that could throw everything, a monkey wrench in everything. And, my grasp is.
It's just.
And I know we can learn nothing by assuming gods and magic. There are a number of things that we can determine about evolutionary ancestry and phylogenies and so forth, but there is nothing that pretending that we were created by a magic spell will ever avail us. There's no future in that. Cindy Lincoln never addressed evolution at all, nor did she give any indication that she even knows what it is, unfortunately. I don't think either of them listened to my opening statement because that was explaining what evolution is. And they're going to say that, and she immediately followed that by saying that evolution is a belief. Everything that I just said about what evolution is demonstrable, measurable, verifiable. Not a bit of it is belief. This is all stuff we can verify. And then if you want to get into what I call the phylogeny challenge, if you're going to argue for a creationism, if you're going to argue that all these animals are not related, then if we get into the taxonomy, you're going to have to show me where the division is, that there isn't one. That's the phylogeny challenge. No creationist will even try to answer this. And the reason is because of difference in our perspectives. If you're a science-minded person, then you look at this as an investigation. It doesn't matter what the end result is. You just want to find out whatever's true, whatever it is.
Good job, Aaron. But I want to get to what the other people are
From a belief the evidence is wrong. They would, because numerous Caesar claims. Look at even know about hailed for all actions that heard same thing.
Damn, bro. Go easy on them.
Gorilla's concept of it. Leave.
Two subjects on the table and you're about to do a third and my brain can't handle all that. Would you for it? It was just obvious looking at those trees that
I mean, this isn't Sal, but I think she's going to bring up some of the same stuff.
so different. Let me see if I can.
Why do we have Rudolph Virchow?
Are you trying to find it? Thank you, honey. That they are so unique. They defy a common ancestor. I would say that, and this is what I was saying, Sal would disagree with me on. I would say many of the families are so different and so unique. They defy somebody saying they have a common ancestor. For me, it was just obvious looking at those trees that, you know, yeah, you could say all the cat family are related, but you can't say a cat's related to a giraffe. You might be able to, and they're all made of cells, obviously. And another thing Sal might disagree with me on is, I don't believe it has to be provable that God made all of us have a nervous system, a digestive system, so that we could study it medically. That's a good idea, but I don't think it has to be proven in order to defy evolution. Personally, I think it makes sense that we all have arms and legs and mouths because we all have to walk. We all have to pick up our food. We all have to live on the same planet. We all have to breathe the same air. So for me, that makes sense. And
if we get to they're going to say the only time we should only do it is purely. I don't even show
yeah,
grocery
that is
first on leap on predations and they were able.
All right, come on, Sal, say something.
oil that is smashed up dead things by this.
The Permian never ever happened. If they're really,
in Permian layers, thrapsid animals that are
still being shoved up an inch or so a year into the Himalayan mountains, that used to be seafloor. If you want to argue for creationism, then you are the one who
problems you have to solve is
right.
Yes,
no,
it is a red
herring.
herring.
And does that show where the taxonomy breaks down?
It breaks down at the cellular level.
Okay, so
It's
you're
true,
saying that God
cellular
had a
level.
cell on the Noah's Ark?
I'm saying that just like those cell families are so unique, that's what we see in the animal kingdom as
Okay,
well.
so did God just create a bunch of cells? And then they become the different animal lineages?
No, the
Are
thing
you
is,
using a red herring?
yes,
No,
it
I'm
is a
not.
red herring.
that's a reason why.
Please.
No, it's a red herring.
Let's
I
agree
told you
not
why.
to do Calvinisms, okay?
Yep.
That's essentially
What I
what
said is it's moot.
believe, right? They believe that God maybe intervened and created life, and then
Wait, I'm sorry.
They're
Can
saying-
you do that again?
They're saying
You
that
don't
on
have
Noah's Ark,
proof.
that Noah had a bunch of cells.
Yeah.
We don't have
Don't
the
put words in my
archetype that
mouth.
begins, I don't know, cats, or is it carnivore, or is it Phalloidea?
Yeah.
Where's the root of all of these claims? And she just said cells.
Those are irrelevant questions relative to the problem itself.
Directly
You don't have
pertinent.
eukaryotic evolution. You don't
If
have
you're gonna
evolution.
argue-
I
if
have
you're gonna
put
argue that all cats are
forward
not
problems
related.
you have to solve as
If you're gonna
evolutionists
argue
if you're going to
that
say it
cats-
evolved.
One sec, just to be sure that
You're
we
gonna
hear
argue
each
that
side.
cats
We'll break it into
and
2 minutes.
giraffes,
We'll
for
do 1 minute
example-
responses. Go ahead, Aaron, 1 minute.
If you're
You
going to
might
argue
need
that
to move
cats
buttons.
and dogs
All
or
right.
giraffes or
That's it.
whatever, wherever the category is that you want to put the divider, if you want to say that
I mean, for Sal, that's like saying if you can explain Mercury's orbit, then you don't have gravity, because classical, the original, like I don't know if you call the original gravitational equations classical mechanics, or if that's just F equals ma and stuff like that. But you take what we know and what has been predicted and what has been successful, and then you say, well, at a finer grained analysis, we don't know this thing, therefore the entire theory is not false, but you can't claim that it's true. That Mercury's orbit is, I think, a perfect analog for that.
there is a divider, you have to show me the justification for your claim. Where is it that the taxonomy closes down? So the specific things to look at Noah's Ark. What did Noah have on his ark that was the archetype to begin what claim?
I don't follow
I don't know.
that question, but
No, it's actually not a difficult. Well, Aaron is. I mean, I know what he means because I've like studied this stuff, but
everybody knows there are different families, and even you would say one of the
praise him.
categories is family.
Yeah, and we are in the ape family. Specifically, we are in the great ape family.
That's your opinion.
That's not an opinion. That's demonstrated.
There's some
It's been organized over
morphological and genetic
centuries.
similarities, but there are some
That's how you determine what goes in what clade.
differences.
Not just similarities
Don't
that are
cross lines.
definitive, that define us as great apes. And we are worse than that. We are in the taxonomic superfamily of Hominoidea, which is the larger classification of apes. That's a fact. That's not an opinion. So you want to show me where the taxonomy breaks and see how far down it goes.
Oh,
And show me
man.
what the divider is.
What are the comments on this? Let's not do Kentovendisms. Also, Cindy, show me where a cell became a monkey. Cindy's only argument is I disagree, and Sal's only argument is look over here instead. That's actually incredibly that's very succinct and very well put. Sal admitting that he wasn't allowed to publish in a biology journal and had to publish in a math journal was priceless. Yikes. Are dogs still not cells? I feel like that's coming up soon. Let me see if I can find that part real quick. This is exactly what flat earthers like Whitsitt do about gravity. They claim Newton replaced that, so I assume Newton is wrong. Yeah. Oh, I got a guest hanging out. Misinformation. Hello, can you hear me?
Yes, can you hear me?
Yeah, I can hear you. What's up?
Good. I saw some of your debates. I value your logic. Can I just see with you, or do you want questions about young creationists necessarily?
You can ask me whatever you want, but just keep it relatively short, I guess.
So relatively short, I encounter the joint connection, let's say, between flat earthers and young creationists when it comes to circular reasoning, I would say. How do you avoid going with them into, because they use this fallacy a lot.
What I didn't follow that.
Circular, how it's called? Circular reasoning, you know?
Yeah. You're asking me how do I avoid it? Or
Yes,
how do I...
when you're presented with it from a young creationist, let's say, how do you move the conversation forward?
Oh, I just point out that they're making the fallacious argument. And I try to give examples. But I just point out, you're just saying it's true because it's true. Or you're saying it's impossible because it's impossible, therefore it's impossible. I guess you just have to have practice with it so you can diagnose it in real time quickly, but yeah.
How do you avoid going to the definition of science when the people in front of you clearly don't understand science and trying to reduce it to the scientific method?
How do I avoid it? I don't avoid it. I have no problems talking about science and how the scientific method works.
When you have, I don't know, someone like Sal, someone like Flatzoid.
What about
you know
them?
where I'm getting?
No, I don't.
He has his own version of science.
Oh, I mean, I don't know. I usually just ask these people, what is science and how does it work? Or I go over like very simple examples of like trying to solve a crime to demonstrate that, yes, we do find proof, or we prove things beyond reasonable doubt. And that's as far as science goes. And we all agree what's reasonable. Even the science denier agrees what's reasonable as long as it doesn't conflict with their religion. But when it does, then all of a sudden they have a completely different standard about what is and is not reasonable. So I just like to point that out.
Thank you.
You're welcome. Anyways, okay, I found the timestamp for it. People said it was at the 50-minute mark. So we're going to, or about 51-minute mark. So I want to reverse a little bit.
And change your mind forever.
I'm sorry, I don't believe you.
I know you don't believe it. Let me prove it.
Honey, I know
mutual exchange.
and it drives me nuts. But cells produce cells, bacteria produce bacteria. I'm sorry, but it's
and that's according to the laws of evolution. There's two laws of evolution that imply that: the law of monophyly and the law of biodiversity, which your husband never understood. I constantly corrected him, and he kept saying this idiotic thing. Well, dogs produce dogs. They don't produce a non-dog. Yeah, you idiot, because that's what evolution allows, right? You cannot grow out of your ancestry.
Okay.
So
Wait, is that one of Kent's ex-wives? No way. I think it is. Dude, that's insane. Aaron has no chill.
never stop being whatever your.
But do you understand how contradictory that is?
If you don't understand what evolution
dog
is,
always
and I
stays
know
a
that you
dog.
don't.
If you're going to say a cell becomes a dog.
What?
I never understand your position. It seems contradictory to me to say that a dog is always going to be a dog, but a cell is going to, over millions of years, turn into a dog.
You mean dogs are not still cells?
That's playing games.
Have you ever heard of the cell theory of biology? You mentioned Virchow. He's the one that came up with that.
So, Cindy, if I can interject a little bit, I feel like
whatever. Are dogs not still cells? What a brilliant thing to say.
like you're asking origins of multicellularity. So
okay, back to where we
our
were.
model, right? And I agree with Aaron that we don't believe things. We have models.
See how far down it goes. And show me what the divider is. Show me what two things that scientists say, well, these two are related to the common ancestors here, and you'll say no, that there's no, they're independent creations. Show me where that happened.
Apes and humans. Show
we
me
are apes.
Common ancestor that
we are apes.
Show it to me.
Look it up. Google it. What is hominoid?
Show me how a cell turns into a monkey.
You just changed the question. You just changed the question again. So
well,
again, we go back to
well, you start off as a zygote and then
what, hon?
No one's claiming the cells turn into monkeys.
What? That's what you believe. That's what the whole.
No, you're not going to put a cell in a dish and then have a monkey jump out of it. That is not the claim. The claim is that there's been billions of years of life on Earth, that for billions of those years, we had single-celled organisms where we don't have a fossil record of, but what your table shows, the tree that you had from Smithsonian, basically, if you look at the different branches.
I don't know. If Sal tries doing this incredulity stuff, should I just ask him, where Jesus's birthmark was? And then if he doesn't know, I'll just say, well, Jesus is God, but we've just proven that God doesn't even exist. If you can't give me the birthmark, then you have no theory. I'm going to write that down. There, I'm going to write that down. Where was Jesus's birthmark?
Branches, you get to the eukaryotic organisms and you have three major branches: the fungi, the animals, and the plants, and they're just one long little tiny branch.
Oh, I don't plan on making it a religious debate, although the topic of the debate is intelligent design versus evolution. I mean, I do plan on basically asking, questions like, I don't have this written down, I guess, but at some point, like, how did God do anything? And there won't be an answer to any of that. So it'll be like, so you can't give me even the first reason to believe your claim. All you try to do is say, well, what you believe in is false.
If you look at the vast array of the single cellular organisms that are out there. Now, this is getting way out of my specialty. I'm a neuroscientist. I study animals, but I know that there's a huge range of genetic variability we can find across a wide range of unicellular organisms, billions of years, allowing for, and abiogenesis is a tough question. I'm not going to say that it is.
Human. This is a topoisomerase enzyme. It is homodimeric. I published on this enzyme. We estimate 60 some interface points to make this. This is difficult. I ask evolutionists how this evolved. They just give me these circularly reasoned phylogenetic explanations. It has nothing to do with taxonomy, but we would be dead without this. It also has to have reformatting in the nuclear localizations and a lot of other things in the prokaryote to eukaryote transition. There are other things with proteins that people seem to not ignore. That's one of my co-authors. published in Nature. And we'll skip this. We can talk about topoisomerases. This is the phylogeny. It's a mess. And I have 1 minute to go. One of the problems in protein architecture is the fit. There needs to be proper charge distributions and shaping so we can get these induced fits. Delving into taxonomy doesn't solve this. I ask evolutionists all the time, how does this evolve? They say, we don't know. We have just a phylogenetic thing. It's a faith statement. And to say that somehow you look at the fossil record and you can claim these things, these complex systems like this, like the EZH2 can evolve, that's a non sequitur. And until you can answer that, you have a faith statement. That is not science. Thank you.
Okay, I appreciate your passion for the Sal. I am not an expert in topoisomerase for sure. However, I did a little bit of looking into topoisomerase. I'd like to share my screen if I could. 1 second here.
I also have to add that if we say that we don't know something, that's not a faith statement. Not having the answer, but saying that you do have the answer, that's the definition of a faith statement. So Sal saying that he knows that was a god, or that he has reason to believe that that's a god, that's a faith statement. That's not what we're doing. I asked him a question he refuses to answer, so he needs to change the subject.
So this is, when we've touched on model organisms, I made up based on this link below, kind of added to it and changed it a little bit. I made this phylogeny, myself, sort of stealing some of the things and elements from this link below. And this is illustrating some of the, what we know to be the last point when there was a common ancestor between humans and chimps was around 10 million years ago, probably a little bit less than that. For humans and mice, around 90 million years ago. Chicken and frog and zebrafish, much older, hundreds of millions of years ago. And then you get into the invertebrates where genetic difference as well as other things, around 990 million years ago, billion years ago, before the Cambrian explosion. Now, what we know is that organization of like 450 million years ago, we don't find humans in those fossil records. But the other thing is that we see sorting of genes as we go through. And anyone can do this. Anyone can go and look. So you can go and use certain resources that are available from the NCBI database, and Sal, I'm sure you're more than familiar with this. You can do a blast search. So this is topoisomerase 1. I know you were showing us topoisomerase 2, but this is topoisomerase 1, where I blasted the sequence of the human topoisomerase to Pan-Tropodotes, which is the chimp. And lo and behold, it's 100% identical. No difference whatsoever. And here's the sequence. You can see this is the human version. All these little letters represent different amino acids. And then here's the chimp version, 100% similar. Now, when you go to a different animal model, the mouse, now we start to see differences. We see substitutions, we see insertions. As Sal pointed out, topoisomerase is pretty important, does important functions in the cell, but we're seeing mutations and differences that occur. As you go down in phylogenetics, now we see additional differences. Now we're comparing humans to chicken and even more substitutions and insertions and deletions. You get to something like the fruit fly, now we have a huge range of insertions and deletions. And of course, if you can go through, and anyone can do this, you pick your favorite gene. You can go to the NCBI database, type in NCBI gene, type in a gene that you like. You can find the human one, that's usually the first one that shows up. And then it'll say run blast, and you can run it. And that blast means that you can choose to compare that gene to thousands of other genomes, and you can compare it to a chimp genome, then compare it to the frog genome, one of my favorite animals since I work with it. And what you'll see is that percent identity of human genes to, say, chimps is going to be quite high. When you get to mice, it's less. Then chickens, it's less. And frogs and zebrafish, it's less, because we know, and this is going to be the consistent pattern that you're going to find, not only with other genes, but also topoisomerase. It's the same thing. And so if this is something that's...
Yeah, this is probably something that I'll bring up. If he says, I'll just be like, that proves it was aliens. There we go.
Like specially created, that's so critical. Why is it that the amino acid sequence can be as much as over 35% different between humans and the roundworm, and have it still work?
I have the answer.
Should it just be plug and play, right? And why do we even have any differences?
Actually, it's not plug and play.
Okay.
We found. By the way, if you'll indulge me, Dr. Chris,
sure.
Scholarly discussion. Thank you very much for looking into that. I'll confirm for the audience what Dr. Chris presented is what I see as well. So he's an excellent scientist. I earlier mentioned the direct coupling analysis. The differences between creatures enables us to do this protein fold prediction. It's a testable prediction if you remove some of the sets who are not able to do it that indicate design. Now the specialty, we can take human topoisomerase, put it in yeast, topoisomerase 2 alpha, beta, put it in yeast. You cannot do the reverse. We didn't actually do it in our experiment. We went from a yeast to a mouse, thing died. The reason is that there are post-translational modifications on the C-terminal domain that are optimized for the multicellularity and the complexity of the human. So you ask why there are differences. In some cases, it's very species specific. You'll see like on the C-terminal domain, maybe 50 more locations for post-translational modifications. You'd only get maybe 5 to 10 on the yeast. So there is, for organisms that are not very complex, there's a lot of toleration for variability. Also, just because you have a phylogenetic diagram does not imply that it evolved, because without the topoisomerase, one is dead. The other thing is when one is going from the bacterial gyrase form to like the eukaryotic form, it has to go from a tetrameric form to a homodimeric, and then the nuclear localizations need to be inserted. These things are like fatal in the process of the evolutionary transition. Just because we have all these fossils and taxonomies, I'm not trying to say I'm changing the subject because I want to avoid it. I'm just saying it's moot. It doesn't solve this problem, and you can accept that it happened, but you can't do so based on any direct evidence except phylogeny or belief in evolution. Therefore, it's a faith belief.
No, Sal, I didn't show any unicellular topoisomerase there.
Yes.
This is all multicellular. So why are there these differences amongst multicellular organisms? If there's just a designer, like you're not going to find designers that if you go to a factory, they're not going to create new types of little things to put into each like computer. If you're designing computers,
experimental evidence.
Put it in, right?
What did Machine Beard say? Oh, this comment I saw earlier. It demands that people believe humans can identify as another species, and that if you're a bigot, if they don't, they can't do that. So it's not possible for humans to. Let me see. I identify as a frog. It is possible for people to do that.
Yeah, go ahead.
Apologies, Dr. Thompson.
That's fine.
I just wanted to say we have experimental evidence. It has to do with the post-translational modifications. There's some slight things with hydropathic properties that are optimized for the creature. There was someone, I forgot his name, he published on that. So there are some optimizations there, but also I have been studying the post-translational modifications. The difference in amino acids, like the residues for like serine and threonine, will enable different phosphorylation states that will change the conformation of the C-terminal domain, and that may be cell type specific in the way it works. So it's a good question. We know humans are particularly sensitive to changes. That's why you could take a human topoisomerase, dump it in a yeast, the yeast lives. You can't go the reverse. And in fact, the differences even happen in the paralogs of topoisomerase, even in the same human genome. We have the alpha and beta isoforms. And we're wondering, why do we have two? Well, we knock out one, there's sickness. So we have experimental evidence to address some of your claims, not all of them. By the way, thank you again. I didn't mean to interrupt you. My sincere apologies, sir.
Yeah, appreciate it.
I still want to know, I mean, did Noah have every species of dog and every species of bovid and all of that on his ark, or did he have like archetypes and then you have other species evolve from that archetype?
I think I like your answer, Aaron. You thought
Aaron.
Yeah, I'm very sorry.
Need a cattle prod, Aaron.
It's just that I thought of that. I'm so sorry. I'm not trying to.
No worries. But so you accept? Do you accept that? Or do you believe that Noah had every species that exists today?
No.
Or did he have archetypes which then evolved into the different species we have today?
I believe, can't prove, but I think when you were in the debate with Charles Jackson and he pointed out the wolf-like ancestry, I agree with that.
Okay, so you accept evolution happens.
Not from a single cell.
Change. I accept that definition of change happens.
So you
and
accept macroevolution by definition, the emergence of new species, at or above the species level, including the genus level, and sometimes even the family level, according to Kent Hovind.
I never thought of it.
Okay, so you accept
because I don't like the definition.
Not just a belief.
I'll tell you why.
Okay. But you accept that evolution happens and that it's not just a belief.
I accept that some evolution happens, but it doesn't explain all the features of life.
We can go back past to the formation of life, back to the formation of the properties that you want to argue for, but we're talking about evolution today. That's the purpose of the debate. So I just want to get a concession. You accept that evolution happens.
You could identify as a frog, but why would you be some sort of ist or phobe if you don't believe they are literally a frog? I really, really highly doubt that he said that if you don't believe that a human is a frog, then you're a bigot or anything like that. But, what do we normally call it when people hear voices in their heads? Schizophrenic, right? So I have now decided scientifically to classify all religion as a form of schizophrenia. Thank you and goodbye.
So now let's talk about the creationist argument on Noah's Ark. If he didn't have all of the different species, then he had archetypes for the beginnings of the different species. And the creationist argument is that all of taxonomy is correct, but only at the surface. And then they want to cut off some little roots for archetypes. If you're going to make an argument that there was a creator, then you have to come up with the origins of these archetypes. You can't avoid that. The only thing you've come up with so far is that God made cells, but that means that everything from the cell on evolved. If you want to...
Well, I'm not going to lie. This is not instructive. Oh, God. Why does everybody have to play the most unbearably loud and obnoxious metal music for their intro to everything? That's what everything is. Listen to this. Okay, well, then it changed to something a little bit more reasonable toward the end, but yeah.
In fact, the theory of evolution is the model describing the...
We're going to jump into open conversation.
Pre-existing parts, the amount of keep on data.
Sal, I'm not sure if that's it, but we know that it's very difficult to hear you.
Can you hear me now? Do I have to?
Yeah, that's perfect. Yeah.
Crystal clear.
It might be that my microphone. So thank you all for bearing with me, and Mark and Eric, James and AJ. Greetings. I didn't have a chance in my opening to extend my greetings. Thank you very much for being a part of this discussion. I'm going to try to understand your question, Mark. And if I misstated, please correct me.
No problem.
The fundamental problem is some of these major protein families, they just won't exist. It makes no sense for them to exist unless they are all there at once. And I gave the example of the topoisomerase. So yes, you can, the question is really, in evolution on trial, it's not just common descent. That's not, there are lots of people that accept common descent like Michael Behe, but think they're problematic things. And that's what I'm trying to attack. But the thing is, like something like topoisomerase, which looks like it would have to come fully formed for it to work. Or at least even if you co-opt pre-existing parts, the amount of miracles to put together pre-existing parts.
That debate happened years ago, and that's not at all what happened. Dinesh said, look, Matt, if you saw somebody hopping around in the mud and said they identify as it was Toad, by the way, Toad, you'd tell them to get up and stop doing this and live a better life. And Matt's response was, no, actually, I wouldn't do that because I'm not a busybody who gets into other people's business. So 0 out of 10 rage bait.
To make a functioning topoisomerase seems astronomically remote to me, even at an intuitional level. And then as I've worked through some of the math for, like, say, the transition from prokaryote to eukaryote, and they have to do things, they have to reformat things and insert things like nuclear localization signals. Once we get into the cellular and molecular details, rather than just looking at shapes and fossils and, speculating transitionals, when we get down to the actual molecular level, which what Michael Behe did, it starts to become like, well, at some point it looks indistinguishable from miracles. And that's where the discussion, I think, really needs to go. A lot of creationists focus on arguing the fossil record. I don't do that. My research has been at the cellular and molecular level. And so I ended up throwing a lot of terms and concepts that people usually don't hear in this discussion, but that's what I research. So my main claim is you look at the part, it's again kind of like looking at the parts of a car. So you look at the first car.
Oh, geez, it's just the same shit over and over again. Incorrect. I'd love for you to play the clip. Sure, why not? If I can find it. No, I have not seen him throw books around.
There you go. My head. There you go. There you go. Okay. But you said that's what I live for. Every day I wake up and I spend a few hours a day, at least at the time I'm not working or eating, counting the hairs on my beard.
Yeah. Okay.
But let's pursue the example even further, make it more ridiculous so we can see how obvious it is. Let's just say, for example, and I don't mean to be going into the trans territory, but I'm going to go there a little bit. Let's say I were to say, I identify as a toad.
So it was toad, not frog. So it's you 0, me 1.
Okay. Okay. And so what I do is I get up in the morning and I start jumping around like a toad. And for a while, people go, hey, that guy's an eccentric, man. What the hell is he doing? And then I go, listen, it's not just up to me. I'm getting a little annoyed that there are all these haters out there who don't respect my identity as a toad. Disney won't cast me in toad races in their movies. I'm denied rights under the Constitution. All these people look down on me and big jokes when I come, jumping by. Okay. So, and my point is not that I've decided to become a toad. My point is I've always been a toad. I'm just a toad in a human body. Okay. Now.
So he said that they have a human body, so they're not saying that they are a toad. You zero, me too.
And I seriously press this. You would come to me, I think, and say, Dinesh, you know, you are a member of Homo sapiens. You're not actually a toad. This purpose of your life that you have ascribed to yourself is based on some deep psychological disorders. You have very poor self-perception of who you are as a human being. And who you are as a human being is you're a son, you're a brother, you're a graduate of an Ivy League college, and you make movies. You know, go do those things and find a better, a higher purpose in life
does make it
than
sound
jumping
like
around
that.
in a dirty pond.
You think that's what I would come to you and say?
I hope so. Unless you, I hope so. You may.
No.
You might come back.
He's so nervous. Matt, please do what I said you would do. I won this argument in the shower earlier.
You might come join me.
Because first of all, I'm not a busybody who worries about what other people do and how they feel and what their
Oh, wow. That's exactly what I said. So you zero, me three.
views
No,
are.
that's not. It's not.
But also,
that's not.
but also, I try to avoid fallacious arguments. And in fact, you
Where's
are
the
a
fallacy?
Homo sapien, but Homo sapiens include multiple different genders, and the fact that someone's gender identity doesn't match what you think it should be is none of your damn business.
Hold on.
So what did you say? You said if a child identifies as a frog, he'd support that child's identity. He'd support that child's identity. There might be a microscopic kernel of truth in what you're saying there.
Hold on. Okay. So right up to this point, you've been invoking science.
And then they're gonna go into something else. So yeah, I don't know. Nice try. I don't think I'm gonna get much else out of watching this stuff that Sal talks about. Maybe I'd have to. I mean, the thing about this topo miso soup cyasin super saiyan protein, whatever that he's talking about. I mean, the guy pulled it up and it matches perfectly the amount of insertions with phylogenetics. So I don't see how that's nothing other than evidence of evolution.
Created by Carl Benz, and then you look at modern cars that have all these new parts and components, and some of these components are very, very, very.
The design argument, I'm going to add this to my notes. I think I got everything I need. So the thing about arguing for commonly designed things is that commonly designed things, quite a lot of them share almost no parts whatsoever. So to point at the phylogenetics and the protein structure and the genes is to say that that's common design. Like pick a I don't know, let's imagine a Boeing 777 or an Airbus. I want to pick something huge. Let's imagine an Airbus versus a just a plane that you like do crop dusting with. How many parts do they share? Probably zero, right? There may be a couple, like there may be some screws that they share, but overall, almost exactly zero things do they share in common. Now compare an albatross to a hummingbird. How many commonalities and similarities are there? Well, at the genetic level and at the protein level, the things they're actually made of, 90%, 98, 80%, something like that. So this thing about design doesn't predict anything. It's just retroactively fitted in to make excuses, because design doesn't say how much design you should share in common or whatever. But like literally everything we know about design today is no, it doesn't actually work that way or extend in the same way that you think it does. So I don't know. What time is it? We'll watch like 5 more minutes.
Very
See if
sophisticated,
anything
like
good
say
comes up.
Fuel injection versus the carburation. And at the protein level, we see the emergence of these.
We lost you again, Sal. Volume wise.
I can hear you. Can I switch to optimal, or like when we actually examine it in detail, especially for what we call multimeric proteins, that's astronomically improbable. And then also, even when we take the same protein going from a prokaryote, like the bacteria.
God.
That's deep. That's right.
Oh yeah, Santa Claus hasn't talked yet. Fucking, his name is Eric. The number of people named Eric and the number of people with the last name Peterson that are on the wrong side of things is absolutely disgusting for me. It is a real problem. Well, yeah, I don't think there's really anything to get. Like I said, maybe tonight I'll listen to him and Erica having their conversation or something to see if there's, I don't know, any information there. But Sal is permanently confused. Yeah. So I don't know. He may try to bombard me with all these like highly technical molecular things like, well, I studied this and I proved that it's actually impossible. It's like, okay, I have no idea what your paper means, but this is just a distraction from the evidence that we currently have. So, his go-to analogy is the watchmaker. You know what's funny about the watchmaker analogy? Watches evolve. How did we first tell time? By just tracking the sun. And then we started looking at shadows. And then we noticed that, hey, wait a minute, you can use shadows to get a different sort of sense of what time it is. And so we started putting sticks in the ground with rocks around them to track hours. And then we invented sundials. And then from there we went to water clocks. And then from there we went to more analog type clocks. So in other words, watches evolve. Pretty funny. I mean, technically they don't evolve without a design. Well, actually, this is kind of funny. You can say they don't evolve without a designer, but actually, no, the first watches didn't have a designer. You literally just looked at the sun. So from an undesigned thing, there's a guiding process behind it, a person. But yeah, I mean, natural selection can produce complex things. We just know it. Triple points? Yeah, triple points are really cool. If you don't know what it is, look it up. You can get something to be solid, liquid, and gas all at the same time. Maybe you have the gay. Uh-oh. He caught it. Forcing people to believe his delusion. You can't really force anything to on anybody. The gee. Yep. I agree. Well, anyways, I'm going to be joining Deconstruction Zone Justin in 45 minutes here to farm some content for him because I'm going to be leaving the state next week for a week. I have to flee. Trump found out who I am and then I don't like him, so they're going to be looking for me. So I got to flee back to South Dakota, which is much more Republican. So I'm gonna blend in with the enemy over there for a week, and then I'll be back. But I should have videos uploaded every single day still, even though I'll be gone. My dad back home, he doesn't have internet, so I might have to schedule all my uploads before I leave, which will be annoying. But I'll do it because you guys are worth it. Huh, kitty cat? Come here. Come here. Come here. They want to see you. They want to see the kitty. They want to see the kitty. The wonderful kitty cat. I promise she actually looks happy almost all the time. She doesn't have a good... It's almost like she's not photogenic. Some people in real life are really pretty, but they're not photogenic. And then some people in real life are way more photogenic than they are actually good looking in real life. And then some people are neither. Anyways, your cat looks like you. I guess since I have a sweater on. Kitty Cat Peterson. All right. What else? What breed? She's a snowshoe Siamese. That's what she is. She has nice colors. She is really pretty. Yeah. She was like all white, like her ears and her tail were black when she was a baby when we bought her. And then she just got darker. I guess I didn't know that. I didn't know that cats do that, I guess, technically. So now I know. But she's still very pretty, aren't you? All right. So anyways, I'm joining Deconstruction Zone in 45 minutes. So you can watch his live stream on YouTube, or I'll be uploading the videos later. Who knows when. Tonight on the YouTube channel Truth Wanted, I'll be talking. I think I'm technically not supposed to copy and re-upload any of their stuff, so I won't be. So the only way you'll be able to watch that is by tuning into Truth Wanted tonight, and that starts at 7 Central, so 8 p.m. Eastern. And then tomorrow, the Modern Day Debate. So there we go. Finland. Yeah. Finland. Okay. I'm going to actually leave this time. Goodbye. Thank you.